Hello, and welcome to everyone joining us for today's Select Science webinar titled Single-Stain Controls, Using Them Effectively for High-Quality Multicolor Spectral Data. My name is Matilde Marques, I'll be moderating today's presentation, and I'm delighted to be joined by our guest speaker, Anna Longini, Global Scientific Affairs Senior Manager at Sony Biotechnology. Please feel free to submit any questions you may have during the webinar today. To do this, simply head to the engagement window to the right of Auditorium and click to open the Q&A window. During the presentation today, we will also be running a few poll questions. Please keep an eye on for a polls icon. If you could please submit an answer when prompted, it would be greatly appreciated. So, without further delay, I'm delighted to hand over to our speaker, Dr. Anna. I'd like to thank her for presenting to us today.
Thank you for the introduction and hello everyone. Thank you for attending the webinar. I'm excited to be here to discuss the use of single-stain controls in spectral flow cytometry. Before focusing on how to efficiently use single-stain controls, I want to highlight that they have always been fundamental in conventional flow cytometry, where they are used to calculate compensation, allowing us to analyze flow data efficiently. And they hold the same importance in spectral flow cytometry as they provide spectral reference signatures that are essential for accurate spectral unmixing. To illustrate the single-stain control's importance, take this gray curve. It represents the emission spectra of a full stain sample. Without single-stain controls to define the spectral signatures for each fluorochrome, we wouldn't be able to decompose and analyze the data properly. So single-stain controls are the foundation of spectral flow cytometry. Without them, accurate unmixing would not be possible. In this talk, we will go over the key requirements for effective single-stain controls, the challenges, such as matching out fluorescence between positive and negative populations, the use of beads versus cells as controls, and the best practice to ensure accurate unmixing and high-quality data. In conventional flow cytometry, we follow well-established rules, many of which remain applicable in spectral flow cytometry. One key rule is using the exact same fluorochrome that stains the sample. We cannot substitute similar fluorochromes based on their emission peak, as seen with Alexa Fluor 747 and APC. With their maximum emission peak may look similar, they have different spectral shapes. For example, if your sample is stained with APC, the single stain control must also be stained with APC. And here I'm showing what happens when an unmixing matrix is calculated using Alexa Fluor 647 instead of APC. It results in an inaccurate unmixing. So understanding this is vital for ensuring effective controls in our experiments. Running single-stain controls every time you run your samples can be a lot of work, especially when using large panels. One advantage of spectral flow cytometry is the ability to reuse single-stain controls if your instrument supports this feature. On the ID 7000, you can store as many fluorochromes as needed in the spectral reference library. Additionally, you can save different fluorochrome marker combinations. For example, if you are running 3 different panels that all contain BV711, you can store 3 separate BV711 references in the library and use the correct one for each panel. Once fluorochromes are stored in the library, you can acquire samples on different days and reuse single-stain controls for the unmixing. These plots show data from 3 different donor samples acquired on different days, but unmixed using the same stored reference controls. This capability not only enhances efficiency, but also ensures that analyses remain reliable, regardless of when the experiments are performed. Although reusing a spectral reference saves time and resource, we need to be more careful with tandem dyes. As we know, they degraded over time. A practical way to spot unmixing errors is examining n-by-n plots. In this experiment, we have PBMCs stained with 6 antibody panels, including 2 tandem dyes. When looking at the plots with all the fluorochromes versus BV785, we see that the BV785 and BV41 pair show unusual shape, typical of unmixing errors. And here we are seeing the spectral shapes of all single-stand controls, normalized spectrum, that were used to calculate the unmixing metrics. Now let's focus on the spectral shapes of only BV41 and BV785. At first glance, BV41 is barely visible, that it's shown in purple. You can barely see it. But when we separate the two, we can see that BV785 was masking BV41 curve. This happened because BV785 degraded, causing it to emit fluorescence from the donor molecule, that's BV41, even though some fluorescence from the acceptor molecule was still present. Comparing the spectrum of BV41, degraded BV785, and intact BV785, we see that loss of energy transfer from donor to acceptor alters the spectral shape. If the single-stand control contains a different tandem dye than the sample, unmixing will not work correctly. This is evident in these plots. However, when we use the correct intact dye as the control, the data appears as expected. So for best practices, always keep single-stand controls under the same conditions as the samples. A good way to accomplish this is preparing the staining, the single-stand controls and the samples at the same time, using the same buffers and temperature conditions. The second requirement for effective single-stand control with following conventional flow cytometry is that they must be as bright or brighter than the sample. This is because compensation assumes a linear relationship between signal intensity and spillover. This plot shows an example where the sample in red is brighter than the single-stand control in purple. As a result, the sample data is overcompensated. In spectral flow cytometry, though, unmixing is based on the spectral shape, so the signal intensity should not matter. Here, I'm showing 3 different single-stand controls. The histogram peaks represent the negative population, a dim positive population in purple, and a bright positive population in blue. After normalizing their spectrum, we overlay and calculate the correlation, showing that they are essentially identical. Now, when we apply an unmixing matrix calculate with either the dim positive control or the bright positive control, the results are comparable. This flexibility allows us to choose the most suitable controls. On the ID 7000, you do not need to worry about single-stand control intensity, as long as a distinct positive population is present. However, always check the manufacturer's recommendation for other instruments. The third requirement for effective single-stand controls relies on matching the autofluorescence of the negative and positive population. As per usual, we gate the positive population, and here this ribbon plot is showing the emission just for the positive population of the single-stand control. And the spectral shape will be a sum of the fluorochrome emission with the autofluorescence emission. This other ribbon plot here is showing the emission of the unstained cells, is the autofluorescence emission. Now, to obtain the fluorochrome spectrum only, the autofluorescence needs to be subtracted. The signal intensity is then normalized, and the spectral reference is ready for unmixing. It's important to remember, though, that the autofluorescence can be impacted by different factors, as the biological state, as for example, differentiation, activation, senescence. And the environment also can influence autofluorescence, as differences in pH and temperature. And we certainly know that buffers and fixatives can affect autofluorescence. Here is just an example comparing the spectral profiles of unfixed lymphocytes and lymphocytes fixed with a fixed lye solution, showing not just differences in intensity, but also in spectral shape. For that reason, as with all single-stain controls, unstained cells should be kept under the same condition as the sample. Again, best practice, prepare the unstained control at the same time, the same conditions as all the other samples and single-stain controls. But what happens when autofluorescence between positive and negative does not match? Looking at those plots, we see a slight but noticeable curvature in the PerCP positive population, indicating some unmixing error. If we examine the spectral shape of the PerCP single-stain controls, we notice these negative values, indicating that the autofluorescence subtracted did not match the positive population. This issue arose because the negative population was gated on cells, while the positive control was prepared using beads. We need to match them. Now, if you have a PerCP that was properly stored in the library using the matching autofluorescence, and we use it as a reference control to calculate the mixing, then the issue is resolved. So what are the challenges in matching autofluorescence? Two main situations come to my mind. When you're using fluorescent proteins expressing cells, sometimes the mock cells are not available, or the cells that express the fluorescent protein and the mock cells, they don't have the same autofluorescence because they are in different conditions. Another situation that can come up is when we have different cell types with heterogeneous autofluorescence. This is the case when we are using more complex samples like tissue samples or tumor samples that we have different type of cells present, and it's not that easy to gate them just using forward and side scatter, as it is easy with PBMCs that we can easily resolve where the lymphocytes and monocytes are. So what do we do in this case that we cannot easily match autofluorescence? One way to overcome this mismatch of autofluorescence by using capture beads. As long as the spectral shape is consistent between beads and cells, unmixing should not be affected. Here, we compare spectral shapes obtained from beads and cells on the ID 7000 for 4 different fluorochromes. The correlation analysis shows that they are nearly identical. So when we compare mixing results for a 10-color PBMC staining panel using single stain controls, beads, or single stain controls cells to unmix the same sample, we find that they yield comparable results. This further supports the idea that either beads or cells can be used, provided that the spectral properties match the sample condition. What are the pros and cons on using beads versus cells? So beads has several advantages. First is availability, right? As long as you have a bottle in your fridge, then you can use them. They also have minimal autofluorescence, so this problem of matching autofluorescence between positive and negative, it's not an issue when using beads. They are easy to use, they have high reproducibility, they are compatible with mostly all the antibodies. They don't have any biological variability, so we don't need to worry if the cells will be expressing or not. And also, using beads avoid wasting precious samples. And disadvantage that we can think about is just when the beads single stand controls, they don't match exactly the spectral shape on the cells due to biological context. And this is the advantage of using cells, right? They will always represent the biological interaction with dyes. Or the single stand controls, if you're using the same type of cell, same conditions, they should represent exactly what you have in your sample. And they can be more accurate for tandem dyes because of the same reason. Of course, if you have cells available, they are more cost effective. But there are a few disadvantages of using cells as well. First, cells will give you more variability, right? We will always have this concern, will my cell be expressing this marker? So the level of markers expression can vary from day to day, and maybe it's an activation marker that will not be present for the single stand control. Maybe you will not have enough positive events. Sometimes we are staining rare cells and it's not that easy to find the positive population. They can be more time consuming in sample prep because sometimes you need to harvest more cells just because of the single stand controls. If you have many dead cells, this can also affect the results because dead cells can interact with the different dyes and also the autofluorescence will not match. When you have limited sample availability, of course, sometimes you cannot afford to have single stand controls prepared with cells. And as we discussed, they have a variable autofluorescence and sometimes it's very difficult to overcome. So understanding these trade-offs help us to choose the best controls for each experiment. Let's just discuss a little bit some best practices during acquisition and validation that can give us more effective single stand controls. First thing is to monitor the instrument settings, right? We need to make sure the instrument is performing well. And we need to regularly calibrate and acquire our quality controls. Use consistent laser and settings configuration. This is very important that you certify that the lasers are on. All the lasers that you're using to acquire your sample should be used to acquire your single stand controls. Confirm fluorescence signal. Ensure each single stand control produces a clear single positive population. Check if you have enough events for both positive and negative populations. And also always check the spectral shape. And how we identify and address these issues. After a mixing, check the end-by-end plots for the full stained samples. And if you spot any issue, check controls and sample quality. Restain or prepare fresh controls if quality is compromised. As key takeaways, always use the same fluorochrome as your sample for accuracy. Ensure autofluorescence matches between positive and negative controls. Maintain matched preparation conditions for reliable mixing. Minimize light exposure, especially with tandem dyes. And when using the ID7000, remember that the brightness of the single stun controls does not need to match the sample. Choose the right material, beads or cells, based on sample availability and biological context. I want to acknowledge the contribution of my colleagues, include Cody, Larry, and the SONYWebina and marketing teams, as well as the Select Science team. And as we conclude, I thank you for your attention. I will be happy to take questions. Thank you.
Thank you, Anna, for that interesting presentation. I hope that everyone listening enjoyed the webinar and found it informative. We will now move on to the last part of today's webinar, the Q&A session. As a reminder, to ask a question, you just need to simply head to the engagement window to the right of Auditorium and click to open the Q&A window. So, to kick off this final portion of today's event, the first question I have for Anna is, I have been using Spectral and I was told that single stain controls should be as bright or brighter than the sample, but you said that they can be dimmer. Now I'm confused. Can you explain again?
Oh, absolutely. When using conventional flow cytometry, single stain controls should be as bright or brighter than the samples, because compensation assumes a linear relationship between fluorescence intensity and spillover. However, in spectral flow cytometry, the mixing relies on the spectral shape rather than intensity. While brighter controls, they can provide clearer spectral signatures. The SONY7000 does not require the single stain controls to be as bright as the sample, as long, of course, you have a spectral shape that's well defined. So the key is to ensure that the reference spectra are accurate, clean, and representative of the fluorochrome used in your panel. Now, if you're using other spectral platforms than ID7000, you should follow the manufacturer recommendations.
Lovely. Thank you, Anna. The next question I have for you is, if I have heterogeneous populations in my sample, should I gate a specific population to serve as the negative control?
Yes. The best practice is to choose the negative population that best matches the autofluorescence of your positive population. For example, if you have a mix of lymphocytes and monocytes and your marker stains monocytes, then monocytes should serve as the negative reference. You can gate them based on foreign site scatter. This prevents the mixing errors caused by mismatched autofluorescence, which can distort your spectral references.
Thank you. So if I spot the mixing errors, but my single stain controls all look fine, what should I do?
Oh, this is a great question. If all looks good and you spot errors, it means the spectral shapes from controls are not matching the ones on your sample. So first thing that happened to me before is to check if all the lasers used for the single stain controls are on when acquiring the sample. So if you have some lasers that are on for the samples and some for the single stain controls, of course, you're not going to have the same excitation and not the same emission. But sometimes it's a sample issue. If the sample was not kept under the optimal conditions, or you forgot, for example, to add polymer stain buffers when using more than one polymer-based fluorochromes, such as BVs, BUVs, etc., that could change the shape of the spectrum as well. If the problem is the sample, you should consider preparing another one if possible.
Great, thank you. And I think we still have time for one more. So what happens if I acquire my sample at different voltages than the single stain controls stored in my library? Can I still use them?
Oh, this is an excellent question, and I'm glad someone brought this up. Oh, the answer is, it depends on the instrument. What I can say is that on the ID 7000, the system adjusts spectral data across different voltage settings, allowing you to reuse stored spectral reference even if your sample is acquired at different voltages. If you are using a storage reference, also make sure that you validate them periodically, just to make sure that the single controls are still representing the same spectrum that you have on your samples, and this will for sure to ensure consistency.
Thank you so much, Anna. I'm afraid that is all we have time for today. I'd like to thank Anna for today's discussion and presentation and for this lovely Q&A session. And thank you to everyone joining us online for submitting your questions. We hope that you have found this a worthwhile session. If you didn't quite get to your question today, we'll follow up with answers to these after the webinar. And if you have any other questions after today's session, please feel free to email myself at editor@selectscience.net. If you are interested in receiving a certificate of attendance, this will be shared with everyone who has attended live today via email a few days after the event. And if you'd like to listen again to today's webinar or invite a friend to do so, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.